Thanks for joining us for another edition of Life Sciences and Biotech CEOs, the podcast focusing on exciting personalities in the life sciences and biotech. Our guest today is Abdulkader Rahmo, co-founder, president, and chief scientific officer of SMSbiotech. SMS is a company building a proprietary platform to use stem cells to attack COPD. AB, as he's known to his friends, and we'll call him here, received his PhD in biochemistry from USC and did his postdoc work at a major lab in Munich, Germany. He grew up as a youth in Damascus, Syria, before departing to continue his education in Switzerland and here in the US. After his education, he returned to Syria and served on the High Council for Scientific Research there. And then he came back to America to start SMSbiotech. I believe I have that resume right, AB.
Yes, it's all right. Thank you, Timothy, for this introduction.
I love the personal stories around biotech. And one of our missions here is to help others enjoy and be inspired by those stories as well. AB, you had a rather circuitous route to the United States here. Can you tell us about how it happened? What kind of was your life story?
You mean coming to the United States?
Yes, from Syria, and what your experiences there were, all that stuff.
I don't know if you are aware of that actually. When I went to Syria, I decided that I will take a road less traveled by. Most people who study in Western university actually continue to work in those societies. But I wanted to see what I can do to elevate some of the technologies there that take care of patients. And indeed, I have introduced a lot of techniques that were not being introduced yet there, and that some of them are molecular biology and cell biology related. So we had quite a success with that. After that, I was invited to hold some academic position, and for me it was important to convey that message to the students there that science is strong, science can change your life, and it can help others. It is crucial and critical for the development of the whole society. I think I saw it as a vehicle for reform. And many of my colleagues did the same thing. It was a collaborative work. Unfortunately, due to the contradictions in the societies succumbed to a civil war that was extremely bloody. I tried to maintain the situation as best I could. At that time, I was heavily involved in research, and I had many students. But there were some personal incidents that definitely pushed me towards leaving the country. I had the intention actually to go into a sabbatical. So I convinced myself it's probably the wise thing to do. At that time, I wanted to explore further the stem cell research that I launched. The reason why I went into stem cells was because there was an immediate need for multi-resistant tuberculosis patients to find a solution that could rescue their life and rescue their lungs. So that was the intent, to find a solution and regenerate the organ, the lung organ. I had presented in Frankfurt and also in the USA, in Florida, and was not sure yet whether I'm gonna continue my academic pursuits, or should I consider establishing a company based on the knowledge that I have accumulated. And my intent was, again, to serve the patient. And that's why I decided to go and establish a company. I thought that this is the faster route towards serving that patient. The offer in Germany was quite enticing, actually. I've been offered a position, a potential position in Max Planck Institute, and it was, you know, a dream position for a scientist. So I had chosen still to build a company. The United States is the best country still for launching a startup, and the support that I found was tremendous. Actually, we have been able to sustain that company for 7 years with mainly private funders. We hope that we move now to the next step and go on into application into human. So we are very close to that stage, and we're looking forward to reach that goal, that original goal of making a difference to the patients.
So what was your experience like working in Syria as one of the lead scientists in the country, which was experiencing, you know, so much turmoil? And the government.
You see, there is an advantage which many people are not aware of when you work in Syria. The fact is, you don't have to worry much about funding for research. So the governments actually funded us quite well and gave us all the money that we want for research. Their salaries are extremely bad, of course. But for people who are really committed to science and are willing to disregard private advantages, you know, of a reasonable salary, we were actually purchasing a lot of equipment and doing and growing quite well. The other problems are, of course, related to the availability of materials and hiring capable people. There are many difficulties associated with the work in science in the third world. However, you can see how a single person can really make a big difference if he is quite passionate and willing to do the sacrifice, the needed sacrifice. And that's probably something general, you know, it's not just bound to the third world or Syria. The regrettable event that came afterwards was related, of course, to the inadequacy of the governing bodies. You know, the situation in Syria is not governed by a democratic system, and people have very little of a say on their future, and that creates huge contradictions, given the fact that many of these young people are aware of how the rest of the world lives. And we made sure that they have the intelligence to acquire the resources about these status of the world. So maybe we actually promoted that situation that ended up so detrimental and so disadvantaged to the society. We have, I think, we have always the struggle between knowledge and reality, between ambition and reality. And we have to understand that the peaceful means of asserting change are extremely important. And whether be it the government or the people, we have to understand the value of exhausting all peaceful means for creating a change and being patient with a change. And then it seems like, you know, being young and or being too much in habit of a powerful situation brought the worst into the society, conflict, and resulted in this sorrowful situation. I hope that the situation in Syria becomes a lesson for people on how civility is fragile and how important for us, especially the intellectuals, is to protect civility, to protect for a peaceful coexistence and productive coexistence between fellow human beings.
Sounds like good advice for America in 2022.
Yeah, there were events where I was also very surprised that indeed there are situations all over the world that are in acute need of such an advice, and they need our involvement. You know, you as a communicator, Timothy, and people like me who lived through these situations, we have the burden of going out to the public, sharing our experience, promoting understanding between people, and enhancing the best in us.
So let's talk for a couple of minutes before we get specific about stem cells in general. Because in preparation for our talk, I've done some reading about stem cells and that sort of thing, and discovered that there are several kinds of stem cells. There are, of course, embryonic stem cells. And one of the things I found that was really exciting was research being done on a single patient only at this point, but where embryonic stem cells were able to reproduce or generate the beta cells that would produce insulin within the pancreas. Wow, right?
Yeah.
There are mesenchymal stem cells. There are pluripotent stem cells, several different names. And yet I think it's safe to say that the research thus far, though it's been exciting in many areas, has often been reached some difficulty in the lab, either because of efficacy, but more so because of ready availability. So maybe what can you tell us generally about where stem cell research was before we get into specifically the single mobile stem cells that SMS is named after?
Yeah. Before we go to the small mobile stem cells, I want to clarify, and definitely assert that there are different types of stem cells. The main three types of stem cells are the embryonic stem cells. These are derived from the embryo, obviously. The adult stem cells, those are taken from the adult tissue from people who are born, after they've been born. Then we have the iPSC, or induced pluripotent stem cells. Those are genetically modified adult cells and made embryonic-like, and have the potency like embryonic cells in differentiating into various cells. Now, the main differences between them are regarding their characteristics. So embryonic and iPSC cells have the advantage of differentiating in many types of cells, like an embryo is supposed to do. They have the disadvantage of when inserted into a human, they could actually generate tumors, because they generate other type of cells which are not suitable to the tissue where they are. We call that teratoma. And there are quite a bit of advances trying to control that to a large degree. It's not clear yet whether we achieved that. The adult stem cells have the advantage of being safe. So they taking from adult, they usually don't form any cancer or encourage the formation of any other cancer in principle. That's what the science so far have shown. But they have the disadvantage of not being able to differentiate in all the types that embryonic stem cells can differentiate into. That means forming many different types of cells. That's what we mean with differentiation. And also in some, when you take adult stem cells so far, and you grow them in the lab, to make enough quantity to introduce into a human, they tend to lose some of their characteristic and potency. So that means you have limited availability, and you need a donor, many donors to satisfy your need of curing patients. Something the embryonic stem cells as a problem don't have, because they proliferate in large quantities, and they maintain usually that potency. So that's the field of stem cells right now. The iPSC are genetically modified, so it's a great discovery. You take a cell, an adult cell, let's say from the skin, and you can revert back that cell to an embryonic state. But once they are in this embryonic state, they harbor the disadvantages and advantages of embryonic cells, in addition to the fact that they are genetically modified. There are now attempts also to obviate some of these disadvantages, but it's still at research level, and we are still not 100% sure whether that has been accomplished. However, we are mature enough at a state where we can dare trying it, like, for example, in producing beta islands, beta cells of pancreatic islands through genetic modifications of the cells into a pluripotent induced cell. Yeah. Now that's the status quo right now. We have chosen a different route. We went into selecting a cell which is kind of quite neglected and overseen by scientists because of their very unconspicuous features. And we found these cells from adult human. We found them to be safe in testing them in animals. We think they are quite safe because they are adult stem cells, although we did not test it yet in the human. We have also been able to grow them to a huge amount without losing their characteristics. So it seems like these cells harbor advantages of the embryonic stem cells without their disadvantages, and advantages of the adult stem cells without their disadvantages. So we got the best of the two worlds without even doing genetic modification. That's why we are so excited about using these stem cells in therapy.
So how did you come upon that discovery? What was it about you that allowed you to investigate something which most others had overlooked?
Yeah, there are several reasons for that. The first reason is I was very open-minded because I was entering a field that is not my specialty. You know, when you are very much experienced in a field, you take many things as a given, which is sometimes bad for you as a scientist.
Okay.
So you have to question the basics, actually. So when I first encountered the cells, it goes against all the textbooks. It goes against everything I know myself. But I had the advantage of telling myself, well, you are not a stem cell expert, so you have to have an open mind and see what happens. So I spent 1.5 years trying to disprove them and try to make them go away. They don't fit any protocols. They don't fit any knowledge before. So I couldn't do that. I spent 1.5 years trying to do that, and I failed, luckily. So the consequence of that is, of course, that you have to pursue it because there may be a big mess here. And there is another reason is I was given enough leverage in determining what I want to research. Some or most investigators, especially in the Western world, are not given this leverage. You know, many of them have to hold on to the grant proposal, to the strict procedure, to the strict agreement, and are very pressed on making publications and spending time for the next grant and the next funding. Some aspects of the research community are actually detrimental to the freedom of research. And I know it seems kind of strange for me to lecture about that, but it's not just my impression. You know, I hear that a lot. Many people have actually abandoned research in academia because of the pressures of that I mentioned, and they chosen another route because of that. So for disruptive technology and for new knowledge, truly revolutionary knowledge, you have to give the researcher enough freedom and enough support to elect how to pursue a groundbreaking research. And I hope that, the community gains that insight and we move on towards, instead of enforcing existing research, just enforcing existing research, giving more leverage to groundbreaking research, to establishing new paradigms, to stimulate disruptive technologies. There is, of course, the will, but I think there is not enough of a mean to make that happen for many, many scientists.
So is anyone else working on SMS cells that you're aware of?
Well, I suspect there is, although I didn't see any significant publications, any publication actually. There may be some publication that looks like they might have a similar cell. I didn't see anything identical, but I have been approached by several postdocs from famous institutions who want actually to do research on that subject matter. That was more recently. Maybe they felt they needed help, with that. Of course, it's a big advantage to have all these years getting familiarized with a single type of cell. So we have a leverage and advantage in that, and I'm sure once a first in human is achieved, there will be a storm of people trying to look into it. Right now, with the data on animals, I'm sure there is a lot of interest in it. The problem for some people is whether they should leave their own research and jump into it. That's the question, they have to answer themselves. And we will see. I'm not afraid of the fact that there will be quite a bit of interest based on what we achieved so far.
I noticed kind of in going through the abstracts for the meeting at Las Vegas, where you made a presentation, that I didn't see any others. Now, I didn't go through it very, very carefully, but I did go through it, but I didn't see any others for SMS.
Oh, yeah, I didn't see it too. And that is, part of building a company, choosing to go the company route instead of the academic route, is to keep secrets and to keep things for yourself, to protect the investors. And I was quite eager to publish because, I have more like an academic mind, but I was advised by the board that it was not the time to publish. We have to wait until later. So we have to be mindful, of course, of the people who have supported that technology. But at some stage, definitely, the spread of this technology can help, potentially help many people. And whether we are the promoter of that or others, I think it will become irrelevant with time. What is relevant truly is that we see that technology taking root and helping people all over the world and making its contribution to resolving such a horrible disease like COPD.
So let's talk now for a minute about actually the experiment that you did on the rat model, how you changed the cells in the lung to replicate an emphysema-like kind of situation, and what the SMS cells were able to do in that rodent.
Yeah. So we selected a model of emphysema, which is equivalent to the destruction of the alveoli, the air sacs. The air sacs are essential for gas exchange. If we don't have air sacs, we would suffocate immediately. So these air sacs deteriorate in COPD, and this is what we call emphysema. In rats, we have a model that can be created by introducing an enzyme that artificially destroys the air sacs in the lung. Of course, we do that in a measured way because if you do too much of that, you will kill the animal. So we had the animal with this emphysema state. We created that animal using the enzyme, and then we introduce the cells through the trachea to the lung directly. And frankly, to my own amazement, we found regeneration and healing in the lung that way exceeded my expectations. So we had, with a single injection of cells, within 1 week, we had 100% regeneration of the alveoli in the lung of these rats. Of course, we introduced human SMS cells into the rat. So my expectation was maybe 20%, maybe 15%. So it way exceeded my expectations. And of course, there were hints towards that from the lab. We have very strong interactions between these SMS cells and lung cells that are key to regeneration, such as the alveolar type 2 cells, a main cell, a progenitor cells, a cell that creates the main components of the air sacs, and a strong interaction with mesenchymal stem cells inside of the one derived from inside the lung. So these in vitro hints were clear, but still, to regenerate the complex structure of an alveoli, an air sac, is really something amazing. And the accomplishment of that in such a short time is very surprising to me. So we have tested even by adding more of the cells, and we're also surprised by the fact that we got the emphysema animals to be better than the control themselves in terms of air sacs. That's also a big surprise to me. So I went and presented that in several conferences. One of them was American Thoracic Society Innovation Summit, and then another one was international the International Stem Cell Research Conference. And the last one was about the longevity therapeutics. I wanted to share that with the world and make sure that the experts are aware of these very surprising and incredible potency for regeneration. So it seems we tapped into a natural phenomenon. Remember, we do not coerce the cells. We do not genetically manipulate the cells. These are natural cells that have been introduced in large numbers into the lung. And they probably do something what's normal to the tissue is. And it's very nice to see yourself enhancing an existing mechanism instead of coercing a natural mechanism. So that's why we got such excellent safety results. So even if we add a huge amount of these cells to the lung of the rats, we never saw any adverse reactions. So we had potency and safety, the best of the two worlds.
And because it is it naturally occurs in the body, is there a different hurdle rate in terms of an FDA clearance to do human testing?
I think that brings us to a challenge, that we have to definitely embrace and take care of. This is an unknown cell, and it's not taken by academia yet, definitely. And it's an isolated phenomenon. The research on that cell is really still isolated, quite isolated. So the FDA now has a burden of very little resources and information on the background, or at least not sufficient information. So we need to educate the FDA with these tests that we've done, the studies we have done, the years of research, so that they are comfortable making a decision on what is still needed. Now, in principle, yes, I think the fact these cannot be denied. This is an adult stem cell that has been isolated from human. We did not change these cells, we just grow them. The cells have been there for quite a while. My first publication was in 2013. And these cells have been tested many times for safety. They have been shown in vitro also to be safe. We tested whether they form tumors or not, and they do not. The fact that they likely to be a natural phenomenon should help the FDA open doors to application in a human more easily. I hope so. I mean, the FDA is really right now besieged with many cell therapy proposals, and I understand the predicament about assigning time and interest, but I'm hopeful that the fact that COPD is unmet, it kills 140,000 people every year in the United States. Let's imagine that number. Many millions are sick. Around the world, it's 3 million dying from that disease. Many people are undiagnosed also, maybe twice as much as, or as much as the ones that are diagnosed. So the situation like that, us trying to address that definitely brings urgency to the table, and also pressures the FDA to act and pressures us to do our best.
So do you have a timeline that you can talk about, or?
Yeah, we have already contacted the FDA regarding what additional studies they need for us to file an IND. So when we receive their feedback, we would be able to determine how many other animal studies or which animal studies is needed. And we have an excellent partner who is very committed to help us with the animal studies, and they luckily are next door to us, very nearby. With their help, we can accomplish these animal studies that are needed quickly, and then file an IND, which is an investigative new drug application. In principle, when you file that, you can start the human testing immediately, so you don't wait for a response. But if your application has some shortcomings, the FDA has the power to stop you, to halt you, and ask for additional testing of some clarification, whatever. So close contact with the FDA and working together will help us avoid any halting or any stopping towards an application to human. The first phase trial will be geared towards safety. So we were gonna bring volunteers, healthy volunteers, and test the cells on them. After that, we're gonna test on sick individuals. Now, the other side of that is that there are plenty of people, some of them we have close contact with, who really wish that process to move quickly and who wish to be among those people that are in trial. But there is so much we can do, you know, to make that happen. We have to adhere, for the sake of this technology and the sake of the future, we have to adhere strictly to the requirements of the regulators.
One of the things I'm always interested in a young company is how the people came together. And it's especially true now for SMS, because I find that perhaps as a result of your having immigrated from Syria to the US and being unencumbered, or maybe it was just because you naturally found yourself intellectually free, unencumbered to investigate the SMS cells, which went against all prior, scientific research. But how people came together. And I was interested because I noted in the board that both co-founders of Massimo are on your board. And here are two guys who started with an idea in a basement or a garage that you could rejigger the way in which oxygen was measured in the blood. And they stuck with it. And today they're a $7 billion company. And in fact, they've become the standard for what we all know now. We see it everywhere, that little thing that you put on your finger to tell you whether or not you have COVID.
Yeah.
So here are two guys who sort of real pioneers, real sort of, leave it all in there sort of people. And then I think about it in terms of where you've come from and where you are today. How did that happen? How did you get together with those fellows?
Yeah, I think I need to divulge a secret here. It's not really a secret. One of the co-founder, which is Mohammed Khair Diab, who is an inventor and a very intelligent person who came with the first idea about that product, that key product of measuring O2 in patients who are moving, especially during intensive care and these type of things, emergency and intensive care. He is a Syrian. So, Mohammed is a friend of my cousin for a long time, and my cousin is a scientist at GPL, Jet Propulsion Laboratory.
Okay.
So that's how the connection came. Mohammed was instrumental to the growth of the company. And he brought also then the CEO of the company, who is his friend, too, very close friend, and they are a co-founder together, which is Joe Kiani, very talented CEO. And he brought with him a lot of support, of course. So there are people who I addressed myself and came also to the company. I think what I can say about most of these investors, they are accomplished professionals. They look definitely into the worthiness of the company, the prospects of the company, and they seek definitely returns on their investment. But it's also a legacy, to be involved in a company that can make a change for many people, health and well-being. Having one sick individuals in a family disrupts the whole family. It's really not just the number of patients, it's the number of families that are affected by that. And I know that your father was affected by that disease, Tim. So I'm not addressing that to you. I'm addressing that to the audience. They have to understand a chronic debilitating disease like COPD really affects the whole family, and we have to find a solution to that. This is a matter of urgency. And some of the supporters that came to us, I would like to mention, for example, Professor Ghassan Kassab and Professor Peter Wagner. Dr. Wagner has been working for decades on that subject matter. He came with a willingness to find a solution for that disease that lingered and resisted all these solutions over all the years. And if the company was supposed to create, some solution for the symptoms or some mildering of symptoms or something like that, I wouldn't have started the company in the first place. This is a company that intends to find a drastic solution, a fundamental solution. And anything less than that, I would consider a failure.
So we've talked also about others who may be involved in the research or maybe not, but I noted that recently Dr. Gary Seek, am I pronouncing his name correctly?
Yes.
Who's the chair of physiology and biomedical engineering at the Mayo Clinic School of Medicine, recently joined SMS as an advisor.
Yes.
So you are acquiring some notoriety, and with that, some additional skill in terms of people helping.
Oh, absolutely. I mean, without these people, without their help and advice, there is no chance we would have arrived to this stage. So Gary is a recent Professor Gary Sick is a recent addition, and I met him at the American Thoracic Society International Conference about 2 months ago. And we had a nice talk. I think he's definitely the person who can bring also the opportunity for applying that treatment or that cell therapy into other areas of other indications. And I'm eager to continue my relationship with him. We see definitely other opportunities looming there for our technology. Of course, I mean, the credo for a startup to be focused. So definitely we would like to be focused on COPD, but once we generate the proof of principle in human, there will be for us a great opportunity to apply that technology in other fields of degenerative diseases. And we have to assess them and prioritize, what type of next move should be pursued. Every pursuit like that takes a lot of time, money, sweat, and a lot of efforts, unimaginable amount of efforts, and psyche. And when I say effort, it's not just you, it's your whole family, the missing vacations, the missing, the missing times together, the missing social visits, the missing all things that you have to sacrifice, you and your family, because of the opportunity that is opening up. So I hope that more people can join us. I recently met also other people who might be helpful for other aspects of these important cells, maybe regarding longevity. We as healthy human being, when we age, we also develop a slightly emphysema. It's subclinical, it's not warranting medicine, but we know that our lungs lose in capacity with time, and we might actually use these cells once they've proven to be completely safe in a human for reason of rejuvenating the lung. And this is a prospect that we actually may need to improve the quality of life of people who are aging, our aging community. Yes.
You mentioned that there are sacrifices that are made by scientists, by anyone who accomplishes great things in this world, and sacrifices to family time and that sort of thing. But I think that it along with that, you need to say, or it needs to be recognized that those family communications that you are able to have while pursuing a passionately held idea, I think probably become better. They become more important, although less frequent. And so you generate a positive impact to your own life, your own daily life, really, in the pursuit of those, passions. And certainly it's for you.
Yeah, I've seen people in different countries who are really suffering in terms of material means and means to develop. Many talents and many thoughtful people disappear in this world or do not appear in the first place because their parents have not the means to nourish that talent, or sometimes not the wisdom to do that. And I think I owe it to society that I have been blessed with the means to develop myself. My parents supported me wholeheartedly during my studies in both phases, undergraduate and graduate studies. They supported me their whole life. They were very supportive. And I owe it to them to make them proud and about me. And I owe it to the society to return some of that blessing to them. So we have to understand that we don't live in a vacuum. We owe every person our well-being. The person who provides us with bread, the person who provides us with a lift, the person who who cleans the street, who help us, who protect us from criminals. We owe these other people our existence, our continuation. And I've seen, what happens when all this support goes away. I've seen it happen in a civil war. You can see the consequences of that. So the consequences of not caring about your fellow human being are dire. They can be detrimental. They can destroy you. We all don't live in an island, and you don't want to live in an island. I refer back to Tom Hanks' movie for that. You can watch that.
Okay. Well, with that wonderful advice, I'd like to close the podcast and thank you very much for your time. And I really enjoyed talking with you in preparation for the podcast as well as in the podcast itself. Thank you very much, AB, and I look forward to following up on your success.
My pleasure. Thank you very much, Timothy, and I wish you success with your mission.
Thank you.